Good morning, everyone. I'm so glad to invite Dr. Karin Hochrainer from Cornell University to present today's Grand Rounds talk. Karin was trained, she obtained PhD from the University of Vienna in Austria. And so then she moved to Cornell as a postdoc and continued to stay there. And she excelled in her work. And so she became an assistant professor of neurology and neuroscience as well. And so her work is mainly on the protein modifications, so the post-translational protein modifications, and how they play a role in either promoting the brain damage or protecting the brain after injuries like stroke. And so today she is particularly going to talk about the ubiquitination, which is the most prevalent post-translation protein modification and its role in stroke. So, Karin, you can start whenever you like. Thank you. I'm going to mute myself.
Thank you so much. Thank you so much, Raghu, for this introduction, and thanks for the invitation to present here today. So I hope that the topic of the talk is not too molecular in a way for this audience. So I'm trying to explain everything hopefully very well so that everybody can follow and understand. So my lab is interested in a post-translational modification called ubiquitin or ubiquitination and its role in ischemic stroke. So in particular, how does ubiquitination, if it does affect the outcome of the ischemia. And of course, this is also all with the idea to, in the end, maybe discover a new therapeutic target that we can use for stroke therapy. So what I'm going to do is I'm going to start out with describing what is ubiquitination. So as I said, ubiquitination is a post-translational modification, and it is like it is like phosphorylation or acetylation or glycosylation, a post-translational modification. It just is a different group of post-translational modification due to the fact that it's an attachment of a polypeptide instead of a chemical group or molecule. And this polypeptide is called ubiquitin, and it has a size of 8 kilodaltons, so it's pretty big. Ubiquitination is ATP dependent, and it's found in all eukaryotic cells. And underscoring its importance, its discovery was rewarded with the Nobel Prize in Chemistry in 2004 to these three gentlemen, Cechanova, Hershko, and Rose. So ubiquitination originally was discovered by these three men by the labs of these three men in the mid-80s. And what it was discovered for is that ubiquitin tags proteins for protein degradation. And what you can see here is the so-called 26S proteasome that's responsible for degradation of these proteins. And it's about 80% of proteins within a cell that will be degraded that way in their lifetime. Now, over the years, it has become clear, though, that ubiquitination does not only lead to protein degradation, but it influences countless cellular events. And how this is possible, I will explain to you in a moment. It is also reversible, so that is important to know, because I'm going to talk about this too in a moment. Now, since ubiquitin is a polypeptide, a protein, it's synthesized by the cell itself. It's can be made from any of these 4 genes that are present within a cell. It's synthesized as a chain and is then cleaved by ubiquitin-specific proteases. And once it's a monomer, it can be attached to target proteins by the sequential action of 3 enzymes. So it's actually pretty complicated how it gets conjugated onto its substrates. Now, what I really want to emphasize and point out is that it can be attached to target proteins in many, many different ways. First of all, so it's always attached to lysins. So this is this K here. and it can be attached as a monomer or a multiple monomer, but it also can form chains. And the way how this is possible is that ubiquitin itself contains 7 internal lysine residues that are depicted here in blue in this three-dimensional structure. And it can use any of these lysines internally to form chains. In addition, it can also make chains via its N-terminal methionine. And the chains can be pretty complicated. It can actually be mixed or branched. So now one can envision, if one looks at that, is that depending on what lysine residue within ubiquitin is used for the chain formation, the chain topology is completely different. So the three-dimensional structure of that ubiquitin chain will look very different to other proteins that are floating around in these different instances. So, for example, the K48 link chain is the prototypical chain that's recognized by the proteasome that is again depicted here. And the proteasome likes this chain to be very globular, so it basically recognizes very globular chains. For example, K63 link chains, they are very linear and they will never be a proteasomal target, and they fulfill very different functions within a cell. So in addition to be degraded by the proteasome and being released, ubiquitin can also be released from substrate via deubiquitinating enzymes without proteolysis. So that is also important to know. Now, why are we interested in this modification in the context of stroke? So I just put in this slide, which I'm really not going to discuss a lot with this audience. I just wanted to say that my lab is working with ischemic stroke, which is about 85% of all strokes that are occurring. And these strokes happen through the occlusion of a blood vessel that can either be thrombotic or embolic. And in terms of treatment, FDA-approved treatments, there is only really reperfusion therapy that aims to reestablish blood flow. So this is very effective, but can only be used in a very limited amount of patients due to different reasons. And there's really no therapies that target brain cells directly to help them or aid them in combating the ischemic stress. So we had the idea of looking into protein modifications because of the fact that if you look on a molecular level, protein synthesis is one of the first things that is affected when cerebral blood flow is reduced. So protein synthesis is shut down, which has been shown in many publications that I've listed here, way before anything else happens. And particularly in the ischemic periphery, where the cells are potentially viable, they have to combat the stress without the possibility of making new proteins. So how does then the cell modify its proteome to be either more stress resistant or to induce apoptosis, anything like that? Well, it will induce post-translational protein modifications. And the reason why we were interested in ubiquitination is that it's notoriously known to be induced with different stressors. I've just put here 3 examples: heat shock, viral infection, or free radical exposure. All 3 cases lead to vast ubiquitination, which is represented by these smears that you can see here, that are representative of many polyubiquitinated proteins. In addition, it's known that this induction in ubiquitination is essential for survival. So if you, for example, delete the polyubiquitin gene, and this has been done in yeast, you lose resistance against, for example, oxidative stress, high temperatures, or starvation. This also happens in mammalian cells, so not only in yeast. So there's, for example, an example of a publication that recently came out in Science by Paul Taylor's group from St. Jude's, where they show that ubiquitination is essential for recovery after heat shock. In light of all of this, we were asking the question, first of all, does ubiquitination change with ischemic stroke? And if so, what are the underlying mechanisms? What are the targets? And what is the impact of the ubiquitination on target function, so that we then in the end can determine, is it protective or detrimental, and can we actually target this in any way for therapy? So I'm going to concentrate on the first four questions today that that we asked first. Before I jump into this, I just want to briefly introduce the stroke models that we're using in the lab. So we do have a mouse model called MCO for middle cerebral artery occlusion, where we use an intravascular filament, so a filament that's introduced into the cerebral arteries to occlude the MCA. And here in this example of laser Doppler flow, you can see this leads to very effective reduction of cerebral blood flow, shown here, and then it remains low for whatever time you would like to keep the filament in. And then once the filament is retracted, you allow for reperfusion. So this is one model we are using, and it's a very popular model actually being used in many labs. The other model that we like to use is in vitro ischemia by oxygen glucose deprivation. And we are using for that not cells at the moment, but actually tissue slices that we isolate from the hippocampus of postnatal mice. These can be then grown on membranes for many, many days, weeks, and then can be exposed to hypoxic chambers and glucose deprivation by just removing glucose from the media. And the good thing about these tissue slices is that you have really an intact slice of tissue rather than just single cells of one type. Now, does ischemic stroke induce protein ubiquitination? And the answer is yes, and we published this like a while ago. So we do find increased ubiquitination in the cortex, which is the ischemic periphery in this model. We do not see that much in the stratum, which is the core. We found that increase in ubiquitination is dependent on reperfusion, and we actually published a paper not long ago showing that the reason for that is just simply that there's not enough ATP in the occlusion period. So now after reperfusion is allowed for, ubiquitination is increased very rapidly as early as 10 to 15 minutes. We show here like 1 hour as a maximum. And it's specifically found on the ipsilateral and not on the contralateral side to the stroke. So now we know now that we have an increase in ubiquitination, but how does this happen? And the first thing we decided to look at here is actually, is the proteolysis by the proteasome affected? The reason why we chose to look at that first is because actually has been shown in some papers that proteasomal activity is slightly reduced after ischemia, including us actually, and then also proteasomal or blockage of proteasomal degradation plays a big role in neurodegeneration and neurodegenerative diseases. So we were interested in looking at that first. So we decided to do that two ways. So first we just looked at proteasomal targets that are naturally present in neurons, which is gluN1 and SHUNK. And we exposed, so this is done in slices here, and we exposed them to OGD reperfusion, and then we checked for levels of these proteins. And as you can see here in the middle lane is that you do see degradation of these two proteins. which can be actually blocked by pharmacological inhibition of the proteasome with a drug called epoxomycin. So what that means is that it looks like that proteasomal degradation is working just fine after OGD and stroke. Another way to investigate that is by using proteasomal reporters. So in this case, we use the GFP, which is usually a very stable protein that it's not really ubiquitinated, but if you attach N-terminally a ubiquitin molecule that cannot be cleaved off, you make it a proteasomal target. And what happens is that that it's degraded by the proteasome in a constant manner, and you will never be able to detect your fluorescence, which you can see here is very dim green fluorescence. And this should accumulate if the proteasome is inhibited enough to lead to a blockage of this degradation. But again, after OGD reperfusion, we do not see any accumulation of signal here. Again, indicating that the proteasome is active enough to lead to degradation of this reporter. Again, we can stop the degradation when we pharmacologically inhibit the proteasome here, as you can see with the fluorescence being higher. And the same thing we find if we use the same strategy in mice. Again, no accumulation of signal after MCO, only when you pharmacologically inhibit the proteasome, and the same we showed with a different reporter as well. So then, what is going on here? How can this ubiquitination be so high after stroke? And in a way, we thought we're going to look next at another class of proteins, which are the deubiquitinases. And the reason for that is that the ubiquitinases have been shown to be reducing their activity after stress. And indeed, what we found really is that after OGD, in this case, we do see a reduction in the ubiquitinase activity that we've shown with two different methods that I'm not going to explain in detail here now, but I think you can appreciate the lower activity here in pink. And this activity reduction we found was actually dependent on free radicals. As we could block or revert that activity reduction by applying MNT-BAP, a free radical scavenger. Also, we find the same amount of reduction if we just add hydroxy peroxide onto the slices, and the same reduction we find in OGD too. So, so that looks like free radicals, which are really there in plenty after ischemia, affect the ubiquitinase activity. And in a way, this all like made sense, because also what we saw is that after ischemia, we have a lot of different ubiquitin chains that we find increased, particularly the K63 chain. That is really not a proteasomal target, as I explained before, and we do see this increase in K63 with with MCO in mice, but we also see it if we apply hydrogen peroxide. We see the same sort of like increase in levels. And what we're doing right now is we are screening for the deubiquitinases that are affected here. So there's about 100 of them in mammalian cells, and we I hope to find something interesting there soon that I can report to you. Now, shifting gears, the next thing we wanted to do is we wanted to identify ubiquitinated proteins after stroke. The way how we approach that is by doing mass spectrometry after ubiquitin enrichment. So in the end, what we did is we took cortical strips of mice that were exposed to MCO 1 hour reperfusion and of sham animals, IPSI and contralateral side. We isolated proteins and then enriched for ubiquitin with an antibody pull down strategy. And then we came up, after a significance analysis, we came up with about 200 proteins that have increased ubiquitination after stroke. About 10 have decreased ubiquitination, and we really don't see any differences in contralateral versus sham samples, which makes sense since there is no induction of ubiquitination on the contralateral side. To assess, to begin to assess where these proteins are localized and expressed, we took our data set and ran it against an RNA-seq database that's publicly available from the McCarroll lab. And they used adult mouse brain for just basically mapping out expression profiles of genes. And what we find when we do that is that our proteins with increased ubiquitination are predominantly found in neurons and not very much in non-neuronal cells. We don't see this enrichment in neuronal cells for the other classes for the decreased ubiquitination and not changed ubiquitination proteins. So then next what we did is we analyzed gene enrichment with gene ontology enrichment analysis, and we found that location-wise, many of the proteins that we found are associated with the postsynaptic density. And we also confirmed that biochemically, showing here by gradient centrifugation. So, I mean, that is very interesting because the postsynaptic density is sort of like the information hub of a neuron, so that receives signals from neighboring neurons, and these signals are going to be processed further to have an output. So if there is anything that disturbs that information flow, then that will be affecting neuronal survival very much. So in order to further confirm this, we looked at PSD-associated proteins, classical PSD-associated proteins, whether they're ubiquitinated, and we found actually many scaffolds to be ubiquitinated present in the postsynaptic density. So one could envision that there is maybe an impact on the overall structure of the postsynaptic density by ubiquitin, and then also receptors like the NMDA receptor, subunits, AMPA receptor, and also the neurotrophin receptor tract B. So also receptor signaling might be influenced by ubiquitination after stroke. Another class of proteins at the postsynaptic density that's very important in transmitting signals further down to the nucleus, so then you have an output of information, are kinases and phosphatases. And this is exactly the class of proteins that we also find highly ubiquitinated after stroke at the postsynaptic density. And here we have a couple of examples that might be familiar to you: CAM kinase, protein kinase C, CDK5, PIK2, creatine kinase, and the phosphatase P10. So by looking at that, then we were wondering, if ubiquitination influences kinase activities, we should see a different phosphorylation pattern after ischemia. So we ran phosphoproteomics analysis, and this is exactly what we find. And then in the end, we were interested in looking further, in particular at the downstream phosphorylation side of these kinases that I just showed you to be ubiquitinated. And what we actually find is that they're really changed and affected with ischemia. So in A, we have PIK2 targets that are generally show increased phosphorylation, indicating a potential upregulation of PIK2 activity. The same is true for CDK5 here in B. On the other hand, protein kinase C and CAM kinase targets appear to be showing downregulated phosphorylation, indicating reduced activity. Now, I will show you data in the following now for PKC and CAM kinase, but we also have already some preliminary data for PIK2, where we implicate ubiquitination in this kinase activity regulation. Well, first of all, matching the downregulation of phosphorylation of downstream targets, we find that both CAM kinase and protein kinase C activities are reduced at the postsynaptic density after ischemia. Now, in order to look whether this is in any way dependent on ubiquitination, we chose a strategy where we treat lysates with a deubiquitinase USP2. So this is a very promiscuous enzyme that will remove all ubiquitin chains from proteins. And as you can see here in this upper plot, this is very effective as the smear disappears, particularly at the postsynaptic density. And now when we do that, what we actually see is that we can restore activities both of CAM kinase and of protein kinase C at the postsynaptic density. And further, if we look in case of CAM kinase, also at the cytosolic and membrane fractions where CAM kinase is also present, we do not see this kind of effect. And this goes in hand with the fact that ubiquitination is really particularly found at the postsynaptic density of this enzyme. So since CAM kinase regulation has been shown to impact, or dysregulation has been shown to impact neuronal death, we decided to look further into that and ask the question, by what mechanism is that actually possible that ubiquitination regulates activity? So we didn't find any, not surprisingly, not any effect on CAM kinase protein levels. So there has to be something else going on. So by mass spectrometry, we found that most of the ubiquitination sites are present in two domains of the CAM kinase, the catalytic and the regulatory domains. And these are very important domains that regulate CAM kinase activity by binding of calmodulin, ATP, and substrate to the enzyme. So we decided to look at that binding of these mediators of activity after stroke. And what we find going in hand with activity reduction of CAM kinase is that calmodulin binding, substrate binding. In this case, we looked at the NMDA receptor, which is a prominent target of CAM kinase. ATP and ATP binding are all reduced after ischemia. And this is, I just want to remind you, all of these binding areas are where ubiquitination is happening. On the other hand, for example, CAM kinase beta binding, which is being executed in the association domain where we do not find ubiquitination, is not affected. So then in the end, again, to look whether this is dependent on ubiquitination, we again use the same strategy with the USP enzyme incubation. And indeed, what we find is that calmodulin binding, for example, is restored when we treat lysates with the ubiquitinase. And so is substrate phosphorylation as seen here. So gluN2B or NMDA receptor phosphorylation is downregulated after ischemia as CAM kinase activity is reduced. This we don't see at the contralateral side, and then it can be restored when USB is added. So in the end, I just want to show you one slide of something that we are doing right now. One thing, obviously, that we're very interested in is assessing whether any of these has any impact on post-ischemic cell death. And in case of CAM kinase, what we did to assess that is we created viruses that knock down endogenous CAM kinase and that overexpress a wild type enzyme of CAM kinase and also a mutant of CAM kinase that cannot be ubiquitinated. So the knockdown virus has a blue fluorescent protein, the other is GFP to detect expression in hippocampal slices that you can see here. And then also the CAM kinase, the overexpressed version is tagged with a flag that you can detect here. So now if we transduce slices with this virus and check for cell death after OGD, what we find is that in case of wild type, you have a certain increase in cell death, and this cell death is reduced in case of the ubiquitin mutant. That would mean that CAM kinase ubiquitination leads to enhanced cell death after ischemia. And if we could somehow prevent that from happening, that could be maybe a potential target. So now, in conclusion for today, I hope that I could convince you that ischemic stroke induces protein ubiquitination, and it does so in the ischemic periphery and particularly in neurons. The increased ubiquitination is caused by a decrease in ubiquitinase activity, not proteasomal degradation, and that tells us that it likely serves regulatory and not proteolytic functions. Then post-ischemic ubiquitination is prevalent at the PSD, where it targets scaffolds, receptors, and kinases, and therefore it is expected to profoundly change protein function at the PSD. And post-ischemic ubiquitination reversibly alters kinase activities at the PSD, and CAM kinase inactivation by ubiquitin promotes post-ischemic cell death. And in the end, what we are looking at right now is which ubiquitinases alter the activity after ischemia and modify outcome. How does ubiquitination impact the architecture of the PSD and kinase activities? And actually, we are looking at what downstream mechanism are implicated in CAM kinase mediated neuronal death. And in the end, I want to acknowledge the people who helped doing this work. So here we have 4 postdocs that I shared with Konstantin Ayadekula's lab at a time, like Kath, Karin, Anya, and Timo, who did many strokes and slices. And then I have 4 very nice, brilliant technicians, Ismarie, Victoria, who both moved on to greater things. And then right now working with me, Emma and Luvna, my collaborators here at Weill Cornell and elsewhere, and my funding sources. Thank you.
Thanks, Karin. That's
Thank
a
you
very
very
nice
much, Karin.
presentation.
Thank
Go
you.
ahead, Dr. Dempsey.
Thank you very much. I think it's very important for us to understand both health and disease are regulated by these sort of proteins and genes, proteins, etc. The more we understand about disease, the more we understand about health, and we move together equally. I think that's really the take-home message I have here. That regulation and homeostasis is really what the body's striving for. Thank you very much.
Yes, definitely. Yeah, the ubiquitination, even for the kinase, we do have data that show that even at normal under normal circumstances, the basic activity regulation seems to be driven somewhat by ubiquitination, which is actually really not known at all. Yeah.
Excellent. Thank you.
Yeah. Any questions? The talk is open for discussion. Karin, let me ask you a question.
Sure.
You are saying that ubiquitination is essential and might be plays a protective role. So it collaborates with other modifications like escalation or simulation, which are also, we know, the same family and also protective.
Yes, that's correct. Yeah. So we and others have shown that both, for example, ubiquitination and simulation are increased after ischemia, and particularly for simulation, it has been shown that it's neuroprotective. So in this case, it's a little bit easier because there's a lot less targets and a lot less involved enzymes, so you can manipulate the system quite well on that scale, which for ubiquitination is impossible so far. But definitely, so it has been shown also that targets that are simulated are ubiquitinated and the other way around. So there could surely be potentially a crosstalk between these modifications after ischemia to modify viability.
Okay, thank you. Guys, any other questions? If not, Karin, thanks a lot again for joining us so early in the morning to give this talk. We appreciate it. next time probably we will invite you to Madison to show you around, and in addition to
Great.
listen to your
Yes.
work. Yeah. Thank you. Have a nice day, Karin. Yeah.
Thank you, Raghu. Thank you, Dr. Dempsey.
Bye.
Bye.
Bye, everyone.
Bye.